Marvelous. So, hello everyone, and welcome or welcome back to the EANA International Spring School on Hydrothermal Vents. This school, which seeks to address these fascinating systems from a number of interdisciplinary science viewpoints in astrobiology, is organized as part of the 20th anniversary celebrations of EANA, the European Astrobiology Network Association. So, after venturing through the worlds of geochemistry and microbiology over the course of the last 2 days, we now turn to the geology and microbial paleontology of hydrothermal systems in deep time. Today's seminar lecture will be given by Dr. Francis Westall from the Exobiology group at the CNRS Centre de Biophysique Moléculaire in Orléans, France. Francis obtained her doctorate in a marine geology at the University of Cape Town, South Africa, and has worked in a number of institutions, including the Alfred Wegener Institute in Bremerhaven, Germany, Nantes University, the University of Bologna, the NASA Johnson Space Center in Houston, and the Lunar and Planetary Institute. She is currently Director of Research and Head of the Astrobiology Group at the CNRS. Francis' research is mainly focused on the geological context for the origins of life and the study of the earliest traces of life on Earth, but she is also heavily involved in the European-Russian ExoMars 2022 mission, of which she is the co-PI of the instrument CLUPI, which is the close-up imager. Francis' presentation today is entitled Hydrothermal Activity and Early Terrestrial Life, and will focus on a number of the topics I've just mentioned. Please, throughout the talk, remember to write your questions for Francis in the chat box, and we shall endeavor to answer as many of these as possible at the end of the presentation. Francis will join us at the end of the lecture for the question session. Wonderful. So on that note, it is my pleasure to give the virtual stage to Francis, and thank you very much.
Good morning, everybody. I'm sorry I'm not here in person to give this talk to you this morning or Wednesday morning. Unfortunately, or fortunately, I don't know. The ExoMars 2022 mission is having its first end-to-end one-day simulation of the mission with the Rover Operations Control Center this week. So as I'm participating in it as one of the simulation officers, I won't be able to give the talk. However, I hope to be able to escape for half an hour to be able to reply to your questions. So that being said, and thanks very much to Barbara Cavalazzi for giving me the opportunity to speaking with you. I am now going to try and, yes, here we are. We have my screen. So I'm going to talk to you about hydrothermal activity on the early Earth and terrestrial life. My talk is going to be in three parts. First of all, I will speak about the environment of the early Earth and the importance of hydrothermal activity, because this is important for life. Then I'll speak about hydrothermal environments and the emergence of life. I know that you had a talk by Mike Russell on Monday. I will be giving a slightly different take on this, just to give you a broader perspective. And then finally, I will talk about the traces of early life associated with hydrothermal environments. Just to put things absolutely straight, we have very, very little data from the early Earth. This is a lovely sketch made by the National Astrobiology Institute of NASA to show the evolution of life through geological time, starting with, yes, I think you can see my arrow, starting with the consolidation of the Earth about 4.56 billion years ago through prebiotic molecules, etc. But in this version, I just want to concentrate on the geology. What do we have from the geology? The oldest physical remains of Earth that we have are zircon crystals, as I'm showing here. These are very, very resistant crystals that can be formed, deposited in sediments, eroded away, redeposited, and so on. And these oldest zircon crystals are dated between 4.4 and 4.2 billion years ago. Also, at this period, we have some geochemical signature of the earliest crust that appears in very early crustal records, rocks, dating back to about 4 billion years ago. The oldest well-preserved sediments date back to only 3.5 billion years ago. Now, this is 1 billion years after the consolidation of the Earth. So in between, we do have some crustal rocks, not very many, but they're highly metamorphosed and highly altered. So with respect to life, well, we don't really know when life appeared because we don't have the rocks. And even if we did have the rocks, I'm not too sure that we would know how to distinguish very primitive life forms anyway. So we think that life could have appeared on Earth as soon as the conditions were convenient, mostly temperature, water temperature, let's say less than 100 degrees centigrade. So somewhere between 4.4 and 4.2 billion years ago. The oldest traces of life, well-preserved traces of life, again, date from these oldest well-preserved sediments, although there is some indication, geochemical indication of traces of life in rocks from Isua, 3.7 billion years ago old. And there has been some suggestion that there may be traces in even older rocks, but I don't think these are very reliable. So this is what we have. The green box represents the rock record that we have. And the pink box is what we have lost. We have no record of the surface of the Earth before about 4 billion years ago. And this is because of two processes, recycling of the early crust of the Earth through mantle recycling, plume tectonics, and also later, as soon as the crust became a little bit harder, the normal plate tectonics that we know from today. So because of this, we have lost the first 500 billion years history of the Earth, and basically the first billion years is not very well preserved. And then, of course, we have to consider the high flux of impacts affecting the Earth. So let's now have a look at the environment of the early Earth after this little preamble. Consolidation of the Earth from planetesimals about 4.56 billion years ago. Then after the Earth had first formed, a first crust, first atmosphere, first volatiles, degassed, etc. The Earth collided with a planet the size of Mars that's being called Theia. There are various estimations as to when this collision took place. I've just put 4.51 billion years ago as one of the estimations. But then impacts continued throughout the Hadean period, throughout the Archean period, gradually decreasing with time. But at the time period that we're going to be talking about, 3.5 billion years ago, they were still pretty high. So let's have a look at the surface of the Earth. On the left, you have here a lovely satellite view of the Earth today, showing South Africa, my birthplace. So subjectively, I'm showing the continent of South Africa. What characterizes the Earth today is the fact that, yes, there's a lot of water, we have a lot of ocean, but we also have a lot of continents. Go back about more than 4 billion years ago. This was not the case. The Earth was basically a water world, and there was very little outstanding or exposed landmass. Again, I'm going to show you what the early land masses looked like. But before I do that, I want to reiterate the concept of mantle plumes and the importance they had on the early Earth, because the early land masses formed above these mantle plumes. So the mantle plumes are extremely hot plumes of mantle that rise because of the heat, and they come in, they're stopped at the crust. But the heat from the mantle head melts the crust, and forms volcanoes, and there's a lot of hydrothermal activity associated with this. Well, and as an example, also an example of what might have the early continents, let's say, or emerged crust on the Earth might have looked like. This is an image of Iceland. It's an image without color because the blue colors that we're so used to today are due to oxygen, and there wasn't oxygen on the early Earth. Now, Iceland, yes, it's forming on a mid-oceanic ridge, but it's forming above a hotspot, a mantle plume. And this is probably what early terrestrial land masses would have looked like. Another, to give you an idea, and especially to emphasize the amount of hydrothermal activity associated with mantle plumes, volcanism, etc. Let's have a look at Yellowstone. This is a 3D, a box model of Yellowstone. You see the topography on the top here, but underneath there's this Yellowstone hotspot with a magma chamber, which one of these days will erupt, but we don't know when. So, and this is another cross-section of the Yellowstone area. You can see the caldera here with the mantle, the magma coming up. But a lot of hotspot activity, geysers, hydrothermal activity associated. Lovely pictures here of geyser activity, and of course the Grand Prismatic pool with which is just absolutely spectacular. So early Earth was hot. Now we are going to jump from today back to 3.5 billion years ago. We're going to go to I'll show you where the ancient crust, well-preserved crust, is preserved later. But first of all, again, I want to show you a cross-section of the Earth's crust, showing the mantle plumes coming up, because there's a phenomenon which is very important associated with them that we see on the early Earth and with which hydrothermal activity is associated. You see that the early crust was very soft. It wasn't as brittle as it is today. There were a lot of faults associated with the bulging of the crust above the mantle, above the hotspot. And these faults, and you can see them in the box model here in red, the cross sections, these faults produced semicircular basins, collapse basins. And this is taken from a sketch by Wout Nijman and his colleagues in an article in Precambrian Research in 2017. So what you see here is shallow water basins around emerged land masses, and the emerged land masses are these orange things. These are volcanoes, felsic volcanoes, and with rivers coming into the sea, and then sort of shallow basins. We will be looking at specifically one of these, well, at the edge of one of these basins, felsic volcanoes with rivers coming into the sea. So we have tidal channels coming into shallow water basins. These environments are also a good analog for the Hadean, when life might have appeared. So this is an artist's view of what the early Earth might have looked like. And I particularly like it because it's quite dramatic. And it's quite dramatic. It shows quite a violent Earth, especially a lot of hydrothermal activity underwater, on the land, impacts, a lot of clouds, etc. So and I will come back to this later, but the early Earth was a very, very different environment to the environment today. It was still very hot, with high flux of heat from the mantle. These mantle plumes were about 200 to 400 degrees hotter than they are today. The crust was softish. There were small sedimentary basins of the order of about 50 to hundreds of kilometers, on top of the bulging crust above the mantle plume. These were like oceanic plateaus. These were the protocontinents, the early Earth's protocontinents, not like today's continents, but protocontinents. There were volcanoes everywhere, a lot of hydrothermal activity and still impacts. So this is a very primitive sketch I made of a cross-section of the Earth's crust showing the volcanoes emerging from the surface of the water, shallow basins, these kind of collapsed basins that Naim was talking about, filled with sediments. And we'll have a look at the geochemical aspect a bit later for the origin of life. Now, there are two areas in the world only where we have really well preserved rocks. These are the Pilbara in northwest Australia and Barberton in the eastern part of South Africa. Throughout my talk, I will be jumping from Australia to Barberton, but I will always show you this map to let you know which continent we're on, because it can get confusing. Anyway, so again, to put things into perspective, this is another primitive sketch that I made to show a box model to show in a shallow basin more precisely the situation. We will have a basalt crust, basalt oceanic sea crust, generally pillow lavas. The volcanoes may be felsic, sort of more evolved volcanoes. There will be hydrothermal vents coming up all over the place. I'm showing them coming up from below the lava from the mantle chamber into the shallow water environments. We have sediments, volcanic sediments. The Earth is only volcanic, so we have only volcanic sediments at the surface of the lavas. There are rivers coming into the shallow sediments. There's a lot of ashfall or tephra, volcanic dust, being blown into the shallow environments during volcanic activity. Meteorites, micrometeorites arriving en masse into the shallow water environments, also on land. So again, I repeat, we have a surface environment that's hot. I'm going to say something here, there's no evidence for ice at all. And the reason why I mention this is because some chemists working prebiotic chemistry, they need to cool, they need ice basically, they need to cool the surfaces down to about 0 degrees so that one of the important sugar molecules for the origin of life, ribose, is stable. But there is absolutely no evidence for this at all. At the rock-water interface, it was hot. And even by 3.5 billion years ago, when the Earth was starting to cool down, here at the interface between the water and the coast, where normally these would be the first places to ice over, we have absolutely no evidence for ice there. Also, the pH would have been extremely variable. It would alkaline, where serpentinites, subcrustal serpentinites are being altered. Acidic at the hot vent sites, neutral in between, quite variable. So, and I will be talking especially about, of course, the hydrothermal vents and seeps, and we'll talk about seeps. As well as the subaerial hydrothermal vents and seeps. So some pretty pictures to give you an idea, put you into the scene. Very hot and acidic black smokers that are typical of deep sea oceanic ridges today. Here you see an image. These are sediments at the bottom of the Aegean Sea, and they look at it. It looks a bit blurred, but the blurriness is because of hot hydrothermal fluids seeping out of the sediments. And this is especially that I'll be talking about, this kind of environment. And then, of course, we have the Geyserites, the subaerial springs, and these are some photographs of subaerial springs from New Zealand. Now, jumping back again, 3.5 billion years. We are now in Australia, and what you see, you are looking at a hillside. You see a hillside with a lot of red. Well, at the top, the red is more or less continuous. These are actually silicified sediments, and they're red because there's a lot of iron in the sediments and they've been oxidized. And then you see these red snakes coming down the hill. And the hill is just basically burnt grass. We're in the Pilbara. Beneath these sediments are basaltic lava flows. We can't see them because actually basalt is very good for vegetation. But the red snakes are hydrothermal chert veins. So they're coming up. There was a magma chamber underneath here. They're coming up from the magma chamber and entering into these sediments. These sediments were deposited in shallow basins. Further evidence of hydrothermal activity. Well, I've shown you the macroscopic evidence, and I'll show you the microscopic evidence. Look at the scale by here, it's 1 centimeter. I actually discovered this by accident. It's a thin section of a rock from South Africa. And you can see a kind of conduit coming up to a surface. We're looking at a cross section of very fine volcanic sediments. The up direction is here. And you can see that this conduit sort of comes up into a funnel shape at a sediment surface. And here's a more detailed view of this. This was a mini hydrothermal vent. So hydrothermal activity at all scales. So, okay, we were in Barberton, just to repeat, we are in Barberton in South Africa at the moment. I want to show you some photographs of the rocks, and then I'll talk about the interpretation of the rocks to give you some idea of the environment. This is the Barberton Greenstone Belt or mountain land, absolutely beautiful area on the border with Swaziland. And the rocks, there was a particular environment that we will be looking at. We'll be looking at rocks from this hillside. You can see a cliff face here, where I nearly lost my finger, but that's another story. Nice close-up of the cliff face. So I will be showing you rocks from this particular exposure. This is one of the rocks. You will see, for those of you who are not geologists, all you see is a striped grey rock. And in fact, it's not just a striped grey rock. It is an absolutely fantastic, magnificent rock because there's a whole history, environmental history here. The black layers are enriched in carbonaceous matter. And this is a cross section. I will show, we are looking at layers and layers of volcanic ash deposited during an eruption, a distal eruption, into coastal waters. To give you an idea, we jump back to today. I want to show you a section through coastal sediments in the Bay of Mont Saint-Michel. You can see here, this is a tidal channel, and you can see the ripple marks on the surfaces of the sediments. Here are the ripple marks. Now, if you dig a trench into this, this is what you see. You see all these crossing layers, dark grey layers crossing each other. This is typical for tidally influenced coastal sediments, this kind of cross-cutting texture. And this is what we are seeing. Oh, and I forgot to mention the early terrestrial sediments, because they were volcanic, they will be black, rather like sediments on Iceland or where any volcanic island. We're not looking at nice white shelly beaches or white quartz beaches. Those didn't exist. So let's come back to these sediments. You can see that they're cross-cutting, sort of with wavy structures. We're looking at a section through the sediments, and these structures tell us that we're in very shallow water. These sediments are tidally influenced, so we're in really, really shallow waters here. And these sediments have also been extremely, in fact, let me go back. These sediments are so well preserved because they have been hydrothermally silicified. Sorry, they have been not necessarily hydrothermally silicified. They've been silicified because the early ocean waters were highly enriched in silica for various reasons. Part of the reason was influx with hydrothermal fluids. Another part of the reason is because influx of highly silicious fluids with rivers. And also, as when the sediments were deposited underwater because they were mostly volcanic, there's a process, a degradation process called devitrification, which increases the quantity of silica in the pore fluids. So these sediments are highly silicified, and that's why they are so well preserved. So let's have a look at the quantity of silica. Here today, modern sediment, well, the quantity of silica varies. Let's say here it's about 50 to 60%. We go back to our nicely banded rock 3.5 billion years ago. The silica content of this sediment is 96%. Now, the same sediment in a very close to hydrothermal activities, very close to a hydrothermal event, looks like this. It's black. It's black, it's black. You can hardly see, you can just vaguely make out some kind of lineations. That this is a hydrothermal fluid conduit, a vein, hydrothermal fluids pushing parallel into the sediments. The silica content here is 99.9%. But it's in this particular area that we have a lot of preservation of early life. Other indications of the hydrothermal influence on the early sediments, if we look at the rare earth elements, the distribution of the rare earth elements normalized to the, I think it's mud of Queensland concentrations, we see a peak of lanthanum, peak of europium, and a peak of yttrium. Sorry, should be here. And these are typical general indicators of hydrothermal seawater or hydrothermal fluid influence. So also, apart from the rare earth elements, the hydrothermal fluids are rich in certain metals, and these metals are concentrated around particles, the volcanic particles, as they are being altered. Here we have a thin section of this banded rock that I showed you earlier, a close-up. You can see a volcanic spherules here. And if we look at this particular area, we can see a concentration of iron, copper, nickel, and arsenic. These are typical metals that are enriched in hydrothermal fluids and that are chelated to the alteration products on the volcanic particles. These alteration products are of variable nature, but often phyllosilicates. Now, I've been showing you some data looking at, well, the rare earth elements data, looking at the bulk signature. We take a portion, a piece of rock, we grind it down, and we analyze it with ICP OES-MS. And this signature gives us a generic information about the bulk rock that represents, pardon me, weeks to months to years, maybe, from a regional to local scale, down to tens of centimeters. And this is work by our former PhD student, Kieran Hickman-Lewis, that came out in Pre-Cambrian Research last year. He's now at the Natural History Museum in London. Now, what he did was he looked, he went on the microscopic scale and used LA-ICP-MS across various biogenic features. This is a microbial phototrophic mat. And he took in situ spot measurements, which gives us information of the order of days and the microscale from centimeters to micrometers. And the information there is quite different. So whereas the bulk information tells us that we have a strong hydrothermal influence, if we look at the ratios of the various REE elements. On the local scale, we get the different elements show us that there's influence also from riverine water, and that on the bulk scale is completely swamped by the general oceanic seawater signature, but not on a local scale. And we can look at it this way again with the spread, the plot of the rare earth elements. We see these nice signatures of lanthanum, europium, and yttrium with the bulk measurements, which gives us hydrothermal influence. But if you look at the spot measurements, we have a pretty flat signature, still a little bit of a signature of hydrothermal activity. But this is typical of material that's been deposited under the influence of terrestrial rivers. So this is really important because we have both a sort of day-to-day record and a longer-term record, both of which are very important for life. Coming back again to this particular diagram that I made, again to show the importance that we also have river input as well as the hydrothermal input. So on what can we say about the early environments on a regional to a local scale? We have water, oceans, lagoons, lakes. They may be deep. We have evidence of material deposited below wave base, but wave base is anything below about 100 meters. Mostly of everything that's preserved, in fact, most of what's preserved is shallow and coastal. We have exposed land, not very much, but there is some, and we will see some lacustrine hot springs from the exposed land. We have hydrothermal vents, subaqueous and subaerial, strong hydrothermal influence, but also terrigenous influence. And on the microscale, we have volcanic particle surfaces, and these will be, I will argue that these are important for the origin of life, and also for as a substrate for life, early life, but varying temperatures, but probably pretty high, variable pH, variable ionic concentrations. Again, these are factors which are important for the origin of life. If we have a look at the global environmental conditions of the early Earth and compare them to today, we can say today a sort of global temperature is averaged temperature is about 15 degrees centigrade. The ocean temperature is about 4 degrees centigrade. pH of the ocean is pretty neutral. The atmosphere is mostly nitrogen and some oxygen, about 21% oxygen. Because of the oxygen, we have an ozone layer. And so this stops UV radiation reaching the surface of the Earth. And according to, I forgot to mention the reference, it's Coquelle and Raven 2004, they estimated that with respect to DNA, we receive about 1 watt per meter square today of radiation. The volcanism is relatively little, hydrothermal activity relatively little, of course, concentrated on the spreading ridges and associated tectonic features, and impacts more or less nothing, okay, apart from the Chicxulub impact that wiped out the dinosaurs. But going back to 3.5 billion years ago, temperatures, especially at the rock-water interface, were well over 50 degrees centigrade. The pH was more acidic. The atmosphere was carbon dioxide. There was very, very little abiotically produced oxygen, less than 0.2% of the present atmospheric levels. Radiation was a lot higher because there was no ozone layer, reaching up to about 100 watts per meter squared. Huge amounts of volcanism, hydrothermal activity, and impacts. So we've got this lovely sketch again without the writing so that you can really appreciate the primordial feeling for the early Earth, how different it was. It was a totally different planet to the planets today. It was an extreme planet. But it was extremely normal for most other planets. Anyway, that's another story. So now I'm going to talk to you about hydrothermal environments and the emergence of life. So, hydrothermal environments and the emergence of life with a slightly different point of view to the one you heard on Monday. If I can get this to move ahead. Yes, here we are. So we have the ingredients of life, water, of course, prebiotic molecules that are of extraterrestrial, preferably of extraterrestrial origin, as well as endogenic production. Sources of energy that included UV radiation, photons, heat, redox reactions, and a suitable geological context. We'll be looking at the geological context here. So let's have a look at the origin of the organic molecules, because this is quite important, and we'll see that hydrothermal aspects are important here as well. So I spoke about in situ origin for the organic molecules. Originally, it was thought that they were produced in a reducing atmosphere, that this level of production is less because the atmosphere is lower, but there was certainly some production of organic molecules during enlightening on the early Earth. Production of small molecules such as ketones, methane, hydrogen, etc., coming from serpentinization processes and exiting through the hydrothermal vents. But the vast majority of molecules, it has been calculated, have come from space in extraterrestrial materials such as carbonaceous chondrites that contain up to about 4% organic matter, and micrometeorites, some of which can be up to 80% organic matter. So these are materials that come from the outer edge of these, outer part of the habitable zone, outer part of the solar system, where such volatiles can condense. Further in, it's too hot for them. They come from the outer part of the early solar system. And so they were delivered to Earth throughout its history to today, in fact, still. They were delivered in the planetesimals, but then degassed into the magma ocean. They were delivered during the moon-forming impact. This is a sketch made by Gruel et al. In Science Advances, showing carbon, nitrogen, sulfur-rich material impacting into the Earth and mixing with the early Earth. And of course, they came in with the meteorites, micrometeorites, asteroids, and so on. And we'll see direct evidence of this in the early sediments. Carbonaceous meteorites are pretty rich in many kinds of molecules. In fact, Schmidt-Koplin and colleagues estimated that there are more than 14,000 molecular compounds, and some people say up to 1 million or more. There are 70 amino acids have been measured in Murchison. All kinds of types of organic molecules are found in these carbonaceous rocky materials. And evidence of impacts on Earth are clear. We have a meteor crater in Arizona. We have one of the oldest meteor domains in impact sites in the world, the Fort Dome in South Africa. It's about 2 billion years old. I think there's an older one that's been found now in Australia with its shatter cones. And we also have direct evidence of the organic molecules coming in, found in the ancient sediments in Barberton. This was a very serendipitous discovery. So coming back again to my sempiternal sketch here, I want to really emphasize the infall of extraterrestrial material. This was going on all the time at a pretty high rate, mixed in with the volcanic ash. We have evidence, direct evidence of impacts in the form of rocks called spherulites or layers of spherulites. You can see this rock here containing a lot of spherules. In this section, they look absolutely exquisite in polarized light. You can see some examples here. But these are all rocky materials. However, in this same sediment that I showed you earlier on, this nicely layered volcanic ash, where the black layers are concentrations of organic matter, we have found some extraterrestrial material. This is the oldest extraterrestrial material, extraterrestrial organic material known in the world, and it's the only example so far. And this was published in 2019 in Cosmochemica Acta, with Didier Gurrier as the first author. So I mentioned that these dark layers are rich in organic matter. And this is a thin section of one of these dark layers, just to show, you can see the dark layer. All of the rest, you can hardly see that they are ghosts of volcanic particles. They've been so highly silicified that you can hardly detect them. In this Raman spectral map, the green represents organic carbon, carbonaceous matter. So this tells you, emphasizes the concentration of carbonaceous matter. And for those of you who are interested, the yellow-orange is silica due to the silicification of the rocks. This light blue here, this is a pyrite, hydrothermal pyrite crystal, and the blue, the darker blue are anatase or rutile portions. Now, I mentioned that these layers have been influenced by tidal currents. The tidal currents mix them up. It's wave action mixing. So again, think about these dark sediments being all mixed. There's organic carbonaceous matter mixed in them. They're all being mixed up. If we look at one of these dark layers, you can see that it's quite broken up here, mixed up. In fact, there's a lot of disruption due to the pressure of fluids coming through, hydrothermal fluids, pore water fluids, again, mixing everything up. And in finer detail, you can see the individual volcanic sediments, the ghosts of the individual volcanic sediments here. Oops. And you will notice in green that they're all outlined by carbonaceous matter, kerogen. So, many years ago, more than 10 years ago now, my colleague Didier Gurrier was in my office, and he happened to see this sample lying on my desk. It came from this rock here. And he asked if he could take it and to study. Now, Didier, he's a physical chemist, and he works with electron paramagnetic resonance spectroscopy. This is a method that analyzes the environment around the outer electrons, and it is capable of distinguishing carbonaceous matter of different origins. Well, this particular lower layer, rich in carbonaceous matter, rich in kerogen, he found a peak which corresponds to a peak that he's seen only in carbonaceous chondrites. He's never seen it in any terrestrial rock, and he's looked at hundreds and hundreds and hundreds of terrestrial rocks. So he phoned me up and he said, "Frances, we have a problem." There's something interesting. So we delved into this problem to try to understand why we were finding such a signature in this rock. And let me talk a little bit about this technique, which was, and this rock, which was studied by Mathilde Bourbin for her PhD thesis, and published in 2013. Now, the electroparamagnetic resonance spectrum consists of various parts. You see a sort of basic line, that the full line here is a Lorentzian curve. And then you can also measure the difference between the peak height, the two peaks, the lower and the higher peak, and the deviation of the line, the measured line, from the Lorentzian, the perfect line. And using these parameters, line shape parameter with peak to peak line width, it's possible to characterize the carbonaceous matter and to determine whether it's of Archean age or younger Proterozoic age, or whether it comes from extraterrestrial materials. And the reason for this is that the organic molecules in these different materials have different compositions and structures related to their origin, and also, especially for the early terrestrial rocks, the Archean rocks, related to the diagenesis and metamorphism of these molecules, which breaks them up and makes them into smaller molecules, aromatic rock molecules. So the carbon generally in this sediment I've been showing you, the general carbon-rich layers plot here, and this particular layer plots here. And but this lower layer, this lower black layer that you can see here, plots in the zone of carbonaceous chondrites. Didier Goury also looked at, found nickel-rich nanospinels associated with this particular layer. And these spinels are only formed when meteors are entering into the atmosphere, and they're formed at very, very high temperatures. They're not found in normal terrestrial rocks. So we have a number of lines of evidence for the presence of extraterrestrial carbon. There is another one that I'll talk about a little bit later on, but for the moment, this is sufficient. Now, this lower layer here, this particular layer, this double layer that we see here, we find all over the locations that we were looking at over a lateral distance of about 2 kilometers. We call it a marker horizon because although it's only about one millimeter or 2 millimeters thick, we find it everywhere. So we have a layer in these sediments comprising extraterrestrial carbonaceous matter. How did it get there? We think that this is probably a distal impact, possibly even dust from micrometeorites. Why was it concentrated in one layer? Probably because the arrival of this material coincided with a pause in eruptive volcanism, and therefore the extraterrestrial carbonaceous matter was not diluted. So it's a very serendipitous event. Everything then was very rapidly silicified and preserved and found by serendipity. Now, the reason why I'm talking about this is because it brings us on to the scenarios for the emergence of life. We know we have organic molecules in the early terrestrial sediments. There have been numerous scenarios proposed for hot spring, for the emergence of life on Earth. The first and foremost are the hydrothermal events proposed by John Barros and I think it's Stephanie Hoffman, 1985, and Mike Russell and all his colleagues have been talking about it since 1997. Martin Brazier proposed pumice rafts as a location because of their porosity. Other people have been proposing volcanic splash pools, Stefan Fox and Henry Strassdeit, for instance. Sabero Springs over the last 4 to 5 years have become highly popular, from David Damer and Martin van Kronendonk and his colleagues. And then finally, and this is what I'll be talking about most, hydrothermal volcanic sediments. This is a mix of the hydrothermal and the sediments. And a couple of years ago, we published a hypothesis paper, or a research paper, also looking at the hydrothermal sedimentary context for the origin of life. We come back to the sketch. We have think in terms of shallow waters filled with volcanic sediments. These volcanic sediments comprise highly reactive surfaces. They come mostly from basaltic materials. They comprise minerals such as pyroxenes, olivines, pyrites, oxides, etc. They are very, very highly reactive, and they're concentrated in this layer. And because of the very high heat flow on the early Earth, there's a huge circulation, hydrothermal flux in the upper crust, with cooler waters coming through into the upper crust, being warmed up, reacting with the basaltic materials, making through serpentinization reactions, coming up in more alkaline immediately into or more acidic, depending on the temperature, into the waters, the shallow basins again. And just a continual circulation, and also continual flow through in these sediments. Again, mentioning variable temperatures, the temperatures coming of the fluids coming up will have been quite high, either low alkaline or mostly higher acidic, but then cooling down. But remember, in these shallow waters, where the crust was very hot, the water's quite shallow, the temperatures at the rock water or sediment water interface were always high. Here we have a little sketch on the bottom left of sediment particles, just to illustrate the flow of these hydrothermal fluids coming through them. Hot fluids at about 150 degrees centigrade at the exits, flowing through, decreasing in temperature with time. The volcanic particles, the sediments, are intermixed with organic compounds, and what we are showing here is the organic compounds with the interacting with these porous materials. The sediments are porous, the surfaces of the particles are reactive, there are organic molecules there. This is a protected environment. We are not affected by UV radiation at the surface. There are all kinds of gradients in these volcanic sediments: temperature, pH, elemental concentrations, ionic concentrations. There is confinement. All the participants, let's say, the ingredients of life, are confined to a small area, which we will see affects their conformation, the way they structure together. They come together to form more complex structures and complexification. So the sketch shows we start off with the organic molecules. We arrive at some kind of protocellular form and maybe some real living cells issuing from these processes with time. One of the important things with the hydrothermal fluids is that, as I mentioned, is the concentration of silica in these sediments and in the seawater. The silica precipitated out as silica gel. So these highly reactive sediments were actually entrained in a kind of silica gel matrix. And the silica gel is very important because molecules chelate onto the surface of the silica gel, and they form a mineral structure, perhaps a mineral membrane through which can aid the prebiotic processes, the chemical reactions, to produce molecules that are of importance for the origin of life. And in fact, one of these experiments that former PhD student Avinash Das made using silica gel showed that the processes at heat under hydrothermal conditions on the surfaces of silica gel led to a concentration of one of the forms of silica, beta furanose, and this is the only sugar molecule that is found in DNA, RNA in life. So let's go back to the Pilbara, because I've spoken to you now about the subaqueous hydrothermal environments. I would like to show you some of the hypotheses regarding subaerial hot spring environments as the origin of life. So here we're in the Pilbara Craton, and this is work from Jorkic et al., where they have found evidence of geyserites in the Pilbara, 3.5 billion years old. And these are some beautiful images of the terracets typical of the geyserites. And also evidence of the subaerial environments are these cracked, hypothesized microbial mats, desiccated microbial mats. So we definitely are in a subaerial environment. And David Damer and David Deemer have hypothesized that it's just in such an environment that life emerged because of the importance of concentrating the molecules. And they have been studying very much the desiccation, processes of desiccation and rehydration in order to get the molecules to fix together. There's and in their papers, they have some wonderful sketches to illustrate this process, with the prebiotic processes occurring in hot springs, and then the material being transported by river processes down to the oceans. As I mentioned, evaporation and rehydration are extremely important to this process for forming micelles or membranous structure. And so they've devised a whole hypothesis from the arrival of the molecules through the hydrothermal hot springs and down into the early oceans. So this is one of the last slides regarding the origin of life. This is my very primitive understanding of how life emerged. So we have our ingredients, the organic molecules, especially the elements, carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. These elements under energy and catalyzers produce something that my prebiotic colleagues, chemists colleagues, call living species, and I think Mike Russell will be talking about this. These are molecules capable of metabolism and information transformation or replication, but within a mineral matrix. And these give rise to a cell with some kind of information processes, proteins, and membranes. And this is a primitive illustration. Now, in all of these processes, minerals are very, very important. And the minerals are important. These minerals come from mafic to ultramafic rocks. They include olivine, pyroxene, potassium feldspar, clays, zeolites, sulfides. They come also from meteorites, serpentinization reactions, and I've tried to show they're important for the concentration of organic molecules, their structural organization. The alteration of these minerals also produces energy and small organic molecules, and they may have some influence also on the chirality of the molecules. Now, this means that no minerals, no life, but I add no rocks and hot water, i.e. hydrothermal activity, no life. So hydrothermal activity is really important here. I'm now going to come on to the third part of my talk, hydrothermal environments and early life. We know from genetic sequencing that the earliest traces of, or the oldest forms of life seem to have been thermophilic. And originally it was thought that maybe this was due to some kind of hydrothermal bottleneck owing to a peak in impacts about 3.9 billion years ago, whereby most of the surface of the Earth would have been destroyed and only organisms in such protected environments could have survived. But we now know that this is not the case. The peak did not occur, and there was just a gradual decrease in impacts. And so this is a real signature. Early life was thermophilic. So let's go back to Barberton. And I'm going to talk again about this, the layer that contains extraterrestrial carbon from these volcanic ashes from the Westall chert. The reason why I'm going to talk about this particular study is because it contains additional evidence for the presence of life at that particular time. So if we look at the high score spectra, this is hyperfine sublevel correlation pulse sequence spectrometry, which measures the influence of certain atoms such as hydrogen, carbon, phosphorus, and silica between each other. We can see, well, we have some examples here from the geological rock record. At 45 million years ago, we see that the hydrogen atom shows a curved signature. The organic molecules contain hydrogen, phosphorus and carbon, of course. The hydrogen has a curved signature, and there's phosphorus. 1.9 billion years ago, at the Garden Flint Formation, we see the same signature. However, 3.5 billion years ago, in the Dresser Formation, the samples I showed, the subaerial geyserite samples I showed you before, we don't see hydrogen. There is no hydrogen signature, but we see the phosphorus, which indicates the presence of biogenic features or life. Now, if we look at the same analysis of a meteorite, a carbonaceous chondrite or GOE, note that the hydrogen atom has a very straight, structure here compared to the curved structure of the younger samples, the younger rocks containing life. There is a very big carbon peak related to the carbonaceous content of the meteorite. Here in the red box, we have two data points from the Westall chert. One of them is the layer containing extraterrestrial carbonaceous matter. Look here, the hydrogen, the shape of the hydrogen peak is straight, just like the extraterrestrial one, but completely different to the curved hydrogen peaks in the terrestrial rocks. And there is also a phosphorus peak, which is not present in the extraterrestrial material. In another sample from carbonaceous matter-rich sample from the same formation, the Westall chert, we don't see the hydrogen peak, but as we don't see it in the Dresser Formation, probably due to metamorphism and the degradation effects of the carbonaceous molecules, but we see the phosphorus. So here in this particular layer, we have evidence of extraterrestrial carbon, carbonaceous matter, and biogenic carbonaceous matter. So let's have a look at this in more detail. Again, I'm going to show the environment, shallow water environment, coastal environment, rivers coming in, a ubiquitous hydrothermal signature in the seawater, also hydrothermal fluids and vents coming up in the shallow water basin. So to give you a taste of what all of this might have looked like, I'm not too sure that we would have seen white smokers. White smokers, although they are highly nutrient rich on the modern Earth, they are typically related to the type of plate tectonics that is typical of the Earth today, that didn't exist on the early Earth. And also, they are pretty low temperature phenomena. On the early Earth, probably the hydrothermal systems would have been higher temperature, also with lots of seepage of these high temperature waters coming up through the sediments. We know that these are extra views of the seafloor showing microbial mats, abundant life associated with all kinds of hydrothermal, low temperature, high temperature systems. And this is due to the fact that these fluids are bringing up nutrients in the form of small organic molecules such as ketones, methane, inorganic nutrients for the chemolithotrophes in the form of hydrogen sulfide, ammonium, and iron 2+. This is another sketch showing the importance of the hydrothermal import of nutrients for chemosynthetic activity associated with these environments. I showed you this image earlier on of hydrothermal vents coming up into the sediments. And I just want to reiterate that there were actual vents in this particular area, as well as the sediments just being completely influenced by hydrothermal fluids. Again, the REE element spectrum underlying the importance of the hydrothermal influence. So if we look at this highly silicified, 99.9% silicified rock, hydrothermally silicified rock, in more detail, we look at one of these black layers. We see a black rock, a matte black rock. It's black because of the microcrystalline nature of the silica, the hydrothermal silica, that has completely penetrated, impermeated the whole rock, the sediments, everything. But it's also black because of the concentration of kerogenous matter or carbonaceous matter. You can still see, although it's been so highly silicified, you can see fine laminations in it. Now, if we take a thin section of this rock, you can see these fine laminations. But note here at the bottom, there seem to be a couple of layers of very dark, spotted rock. And these spots are actually, volcanic particles that are very heavily coated with carbonaceous matter, with kerogen. You can see close-ups here of this matter. If we look, we can use Raman spectral mapping to determine the nature, what the clots really are. And the oval red outline here shows an original volcanic particle. You can hardly see it here, but we know that it existed because of the anatase, the blue anatase here, which is the alteration product of the particle. You can actually just see a little bit of it peeking out underneath this black matter. This black matter is carbonaceous matter, it's kerogen. There's a huge amount of kerogen here. The whole sample is completely permeated with kerogen. Now, if we take a cross section across one of these volcanic particles, you can see the volcanic particle here, and the kerogenous matter, again outlined in green here, coating the volcanic particle. The particles were thickly coated with corrogenous matter, and judging on the thickness here, up to about 30 microns thick. And the coatings are highly irregular. Now, we have interpreted these features as microbial colonies, chemotrophic colonies, just using the volcanic particles as a substrate, possibly chemolithotrophic microorganisms first colonized the volcanic particles, and then as they died off, they would have been colonized by what I call the cannibals, the chemoorganotrophs. But we have more evidence of this, and I'll go into this in the next few slides. Now, if we look at the layer above, it's not quite so dark spotted, but it's still pretty spotted. It comprises stellate clots. These are carbonaceous clots, totally irregular in size and shape. They have formed in an extremely fine-grained silica, what seems to be a silica very fine dust matrix. You can see some very faint laminations here. It's basically a silica gel mixed in with volcanic dust. And the extremely irregular shapes indicate that these features formed in situ. They weren't formed elsewhere and they were deposited. They formed in situ. They grew in situ. We've been using other techniques to try to understand if we have a better signature of biogenicity for these particular structures, because we believe that these are microbial colonies that formed in the silica gel. Just to give you an idea of what this looks like, that we have the impression that this is all organic matter or carbon, but it's not. It's all silica, and the carbon is just in between the silica grains. This is the hydrothermal silica that has permeated everything and precipitated, pushing aside the degraded organic matter. But we can do other analyses of these degraded organic matter. So we did using PIXI spectrometry. This is proton-induced X-ray emission spectroscopy. We can analyze these structures and document. We can document the distribution and quantify the elements associated with them. Now, this gives us a spectrum as such with a blow up of this particular area. But what we're seeing particularly are the typical hydrothermal fluid traces: iron, cobalt, nickel, copper, and also arsenic. We're seeing arsenic here. But if we look at the distribution, the first thing that jumps out at us is the fact that in the middle, there's a splotch, and that splotch in the middle is a volcanic dust particle. It's very small. It's volcanic dust particle. And so this particular structure grew around the volcanic dust particle. But if we do quantification, outside of the area of the volcanic dust particle, and also with the spectral mapping, we can see that there are concentrations of elements, particularly iron, nickel, copper, and arsenic, which are in higher concentration here than they are elsewhere. And we think that this is related to the fact that life processes need these elements for their metalomes. They need these elements for particular compounds that are used in metabolic processes. These are compounds that are used still today by microbial life in their metabolic processes. Probably also there was some chelation of these elements as the organic matter degraded, the colony died, and so the degrading organic matter would have scavenged the elements from the surrounding seawaters as well. So we have evidence of thick colonization, heavy colonization in the vicinity of hydrothermal vents. And not only were we finding chemotrophic microorganisms, but also on the surfaces of sediment layers, some evidence of phototrophic organisms. In this particular example, not very many, because they were completely outweighed by the chemotrophic clots. But on this sediment surface, before there was some further deposition and colonization by the chemotrophs, we find the remnants of phototrophic biofilms. You can see a slightly wavy laminae here. And if we look in more detail, you see that the wavy laminae really coat this sediment surface. These are anastomosing layers. They're about 10 microns thick. They incorporate other particles. They have all the typical characteristics of phototrophic biofilms. Note, we are in an anoxygenic environment, so these would be anoxygenic phototrophs. Now, again, through serendipity, we were able to find one particular layer, find one particular layer of sediment, which had a really superbly preserved microbial biofilm, preserved in 3 dimensions. This is the sediment surface. It's partly, well, the sediment broke open on this particular surface because you can see some kind of filmy structures, layers of filmy structures here, all orientated in sort of fibrous or filamentous, orientated in the same direction. This would have formed a kind of slight layer of weakness, which allowed the silicified sediment to break open on this particular surface. So these layers upon layers, were formed by these are robust filamentous films formed on the sediment layers. They're formed of superimposed layers of biofilms, as we saw in cross section, one 10 micron layer above another. And in this particular example, we see distinct orientation. Now, these features were silicified extremely rapidly. And in this particular case, this sample came from the very highly silicified sediments just above the basalt lava. So definitely very highly influenced by volcanic and hydrothermal activity. Now, the film actually comprises thin filaments. They're not very easy to see because they're thickly coated with polymer. This is extracellular polymeric substances. They thickly coat the filaments, but occasionally we can see a filament. These are consistent. These are not fibrils, these are filaments, and they're less than a micron thick. We can see evidence of formation under some kind of current flow because the filaments are overturned and portions of the biofilm have been torn off. So we're looking at quite a strong flow here. And remember, this rock is 3.33 billion years old. This is the most exquisite preservation. We have evidence that the film was exposed, subaerially exposed with desiccation cracks. You can see the desiccation cracks here and here. And in my hand here, on the bottom right, I show a similar phototrophic biofilm taken from a beach surface with desiccation cracks. So this film formed underwater, but it was more or less at the interface with the water because we have desiccation. And we also have a suite of pseudomorphed evaporite minerals that are embedded within the filamentous films. And from the shapes, we can say that we have aragonite, gypsum, magnesium, calcite, and halite. So because this feature was quite large, we were able to do a lot of analyses. We were able to dig trenches in it to observe its vertical structure. We were able to take thin sections really thin, less than 19 nanometer thick sections for high resolution transmission electron microscope observations, thicker sections for all kinds of analyses such as high resolution scanning electron microscope, nanosims, synchrotron, etc., etc. We had a field day here. So this is one of the sections that we excavated from the microbial biofilm. The bottom is here and the top is here. At the top, we see these minerals, embedded evaporite minerals. There is a silica coat on the top and here on the bottom because it was desiccated, so it was lifted up from the sediment surface. The dark splotches, this is a backscatter scanning electron microscope view. The dark splotches are actually rich in carbonaceous matter. But the bulk of this biofilm is granular carbonate, and I'll show you examples of that later. And we see these tiny floating crystals of quartz embedded in the biofilm, just as one would expect from a sticky phototrophic mat. So I'm going to show you some details of first of all the granular texture. This is actually what preserved the microbial biofilm originally, and also of the remnant kerogenous texture. Now, this is an exquisite example because we are able to go into the detail that you could only hope to do with specific probes on modern microbial mats. But here, because it's so well preserved, totally silicified. I'll show you nanosims scan through the whole of the sediment, just showing that it's totally silicified. We can understand much better about the workings of early phototrophic biofilms associated with hydrothermal activity. So let's have a look first at this granular texture. You can see the granular texture here. It comes out nicely in this high resolution transmission electron microscope image. But especially, this is a study I did with Barbara Cavalazzi, our EANA president. And she was looking at this granular texture and was able to identify very tiny crystallites from 3 to 10 microns in size, and using the high resolution transmission electron microscopy and spectral nature, she was able to identify that these were nanocrystals of aragonite. So if we look at the remnants of the kerogenous material, again, using high resolution transmission electron spectroscopy, we see that it's got an alveolar structure, like a honeycomb. But there are small domains in it, that indicate that this is material, it's kerogenous material that's starting to perhaps become slightly graphitized. So this tells us that we're not looking at a young contaminating phenomenon. We're looking at something that's really old, that really is 3.3 billion years old. Using the synchrotron, this is work done with Loris Laramel and Alex Siminovici, using microzanes at the sulfur K edge. We were able to study two particular areas. One area is the kerogen-rich area, and another area, the carbonate-rich area. The kerogen-rich area shows that we have an organic molecule rich in sulfur, thiophene, and up to 1% sulfur. Now, this is a lot of sulfur, and this is probably due to the process of degradation, diagenesis, and sulfurization. But we also see that there's some sulfate associated with the carbonate. So before I carry on, I'm getting carried away here. Let's have a look at what the nanosims tells us. We take a nanosim scan across the depth of the sample. This is a map of the carbon to nitrogen ratio, showing us that the hotspots where there is carbonaceous material enriched in nitrogen. This is the silica. This is the sulfur, which shows us that there's also sulfur associated with the carbonaceous matter. But the silica is this green one here. It's present the whole way through. The whole of the material has been completely silicified. So we are indeed looking at a beautifully preserved phototrophic microbial biofilm, nearly 3.5 billion years old. Finally, this is work that I did with Volker Thiel. He used TOF SIMS to have a look at the carbonaceous matter associated with this material, and also picked up molecules, evidence for aromatic molecules such as thiophene, but plus all other molecules showing the typical carbon pattern of biogenic matter. So we have, in addition, also the carbonation, the analyses of carbon isotopes, indicating, and this is on the finely layered material that I showed you earlier on, giving distinct indications of isotopic signatures that are very compatible with life processes. Finally, a last study done with Claire, Ronnie, and Bard, but we haven't published. We did an isotopic trail through the SIMS analysis across the section of this section, down this section of the microbial biofilm, and these are the results. So you can see that we very definitely have a structure that is consistent with microbial origin. However, apart from all of these features, we can make some interpretations about the structure. Okay, we have the primary structure was a phototrophic biofilm, but then the evidence of calcification, and the presence of sulfur here, sulfurization, tells us that we're looking at a consortium whereby you have the active phototrophic mat at the top, the photosynthesizers at the top of the biofilm, and below the chemotrophic organisms that are degrading the dead matter produced by the phototrophs. And this is a typical situation of microbial degradation in just such a phototrophic biofilm. And because of the changes in the pH produced by sulfate reducers or sulfur reducing microorganisms, this results in the precipitation of, the release of calcium associated with the EPS, the polymers, and its combination with carbon CO2 dissolved in water to produce the silicification. It was then rapidly fossilized. Now let's have a look at an example. Going back to Australia to have a look at subaerial examples. This subaerial example is again the geyserite from the Pilbara, 3.5 billion year old structure from the Pilbara, and work by Tanya Djorkic and her colleagues has shown that these, as I mentioned, these are very similar to modern geyserite communities, but also that there is plentiful evidence of microbial biofilm formation, microbial mat formation. And just serendipitously, more than 20 years ago, I was visiting the same outcrop and was fortunate enough to see a squashed stromatolite. Stromatolites are quite common in these rocks, but this one is particularly intriguing because you can see the the bent, the squash nature of the soft layers of the stromatolitic shape. So now we're going to stay in the Pilbara because I would like to just show you a contrasting trace of life, and that is early life in an oligotrophic environment. This is a type of environment that is not nutrient rich, and so the development of a microbial mass is a lot lower. So this oligotrophic environment that I'm going to show you again is the tidal channel environment at the edge of a volcano. So remember, we have cross-bedded sediments, layered laminated sediments. What we will be seeing is very similar to this, but nearly 3.5 billion years old. We're going to go to the Kitty's Gap Chert in the Pilbara. This is 3.45 billion years old. Again, the image of the modern sediments on the left, and this is the rock, cross-section of sediments, volcanic ash sediments in the rock. The sample I actually studied was this portion taken here, but we'll be looking at the same kinds of features that are illustrated here in the red rectangle. So here is the sample. What are we looking at? Again, layers of volcanic ash deposited in a very, very shallow environment. If we're in a tidal channel, you can see the cross-bedded sediments here. This white layer actually indicates subaerial exposure. The tidal channel reached exposure. The translucent crystals embedded in this white layer. These are pumice, pieces of pumice grains embedded in the surface here. So, the sort of translucent material cutting through the sediment, semi-lithified sediment here. This is a penicontemporaneous hydrothermal infiltration. So we have volcanic sediments influenced by tidal currents, that were then exposed by regression of the seawater. Then seawater came back again. The land surface was lower. The black and white layers here are airfall volcanic ash. The very black layer here is very, very fine volcanic ash, like something like a silica gel mixed in with volcanic ash. So these are all different kinds of microbial scale habitats, and they were all inhabited. I'm just going to show you a few examples here. Again, this is a thin section of this particular rock. You can see the pumice grain here, the hydrothermal infiltration here, the tidally influenced sediments here. Now, this rock comprises very angular volcanic particles. They're mostly volcanic glass, because the protolith, the original rock the volcanic rock was rhyolitic. So it's full of these very angular clasts. Now we'll have a look at one of these clasts using Raman spectral mapping. Again, here the green is kerogen, the blue is anatase that's formed around the altered. You can see the small spotted structure here. This is anatase around the altered volcanic glass. The pink is the remnant. The volcanic glass was rapidly altered to phyllosilicate, which with metamorphism became muscovite, so it's muscovite. And the red is silica, pervasive silicification. And this is the Raman spectral map showing the maturity of the kerogen that's remained. Now, again, in one of the thin sections of this particular sediment, we noted that some of the particles seem to have tunnels running into them. Now, the light gray here is the volcanic particle The mid-grey is the silica matrix, and these tunnels end at the edge of the volcanic particles, like here and here. Now, these tunnels seem to be filled with some kind of mucus, mucus structure. We hypothesize that these are corrosion tunnels formed by microbial communities that colonize the surfaces of the particles. And I'll show you why we think that this is a likely reason. And again, the presence of this organic polymer, silicified polymer, would support this hypothesis. But let's look at in more detail why we think this. Well, some of the high-resolution scanning electron microscopy analysis of some of these volcanic particles shows that they're coated. They may be coated by what I call a carpet of beads. The scale bar here is 10 microns. The platy minerals here are the muscovite that's replacing the volcanic particle. This is a quartz crystal. This is part of the silica matrix that's up from the silicification. But this carpet of beads is rich in carbonaceous matter. We did some bulk analyses. The bulk carbon isotope signature is about minus 26 per mil. Bulk carbon content is pretty low. It's between 0.01 and 0.1. So let's have a look in more detail at this carpet. You can see the border of this carpet with the phyllosilicates here. And these beads, we see in more detail that the beads seem to be in pairs and two sizes. One size almost 1 micron in size, one size of almost half a micron in size, and they seem to be coated by some kind of film, smooth film. Other details of these features, we can see that occasionally these balls that seem to be in pairs. There seems to be a meniscus between the two pairs, and the surface is quite irregular. And again, we can see two classes of these objects. One, a small one, half a micron in size, and a large one, nearly a micron in size. We interpret these structures as fossil microbes. Now, it's really rare in the early Archean or the Paleoarchean to find preserved individual microfossils. Normally, we just see the generic carbonaceous matter issuing from degraded colonies. But here in this case, we actually see these organisms. So we think we're looking at two species of organisms, because there's one which is definitely larger, one that is definitely smaller. The pairs we think indicates cell division, especially where we see a meniscus here. And with this particular individual, we think we are seeing life and death, because one part of the pair is deflated, and this happens when an organism dies, and it lyses, and its internal matter leaves the cell, and cell deflates. So at 3.5 billion years, we have evidence of life, cell division, and evidence of death. And this, I think, is absolutely incredible. And the reason why I think these cells were particularly well preserved is really because we're looking at an oligotrophic environment. There was not a lot of nutrients here. These organisms, they were floating around. They colonized sediment surfaces. They colonized particle surfaces, but there were not a lot of nutrients to support biomass development. So these organisms, in order to be preserved with such fidelity, they had to be fossilized really quickly. And from experience that we've been doing in my group, we know that fossilization starts within 24 hours. So we really are looking at organisms that probably chemolithotrophs that colonized volcanic particle surfaces, but they were very rapidly silicified, and before they could degrade, although not always. We will see an example here on the left, some other examples of these dividing cells, but here on the right, a chain where the cells are really pretty badly degraded. They've all been lysed, so in this particular instance, the organisms were dead when they were fossilized. So oligotrophic environment, not a huge amount of nutrients, and no chemoorganotrophs to degrade these organisms, which explains their very good fossilization. So I'll finish my talk here. We have looked at hydrothermal activity on the early Earth and its effects on early terrestrial life. First of all, I've shown you how do we know that there was hydrothermal activity on the early Earth, and I've shown the importance of hydrothermal activity on the early Earth, perhaps ad nauseum, but anyway, you get the picture. I've looked at hydrothermal environments and the emergence of life from a different point of view to that would have been shown you by Mike Russell, and looking at the hydrothermal environments on the early Earth and early life, the kinds of the importance of nutrient-rich fluids influencing biomass development close to the hydrothermal vent exits, as opposed to nutrient-poor environments, oligotrophic environments, where there was very little life. So I will end there and take your questions. Thank you very much.
Francis, for a detailed tour through hydrothermal environments on the early Earth, and I'm glad you could join us for the question session. We have a few questions, so I will go through in the order we received them. So the first question is from Marie-Paul Basset, who asks, in relation in the first part of your talk, you give temperatures of 60 to 300 degrees C for acidic hydrothermal vents and 60 to 150 for alkaline hydrothermal vents. How do you justify the use of these values?
This is a good question, Marie-Paul, and obviously the two values are incompatible. It depends on, I mean, there were certainly hot vents, there were certainly acidic vents coming out. The evidence for lower temperature carbonate alteration is there. We see the pillow lava basalts altered to carbonate, so that's obviously lower temperature. But in the shallow water environments that have been preserved, because these are the only environments that are preserved, we don't have any deep water rocks preserved. We see mostly, probably, well, I think it's the interpretations come from the isotopic analyses of oxygen and silicon, which are tending to show hot environments. I suppose it depends on what you mean by hot. From a microbial point of view, anything over 80 degrees, 100 degrees is quite hot. So from sort of the hot black smoker point of view, it's not very hot. So thank you for pulling this out. This was incorrect. So thank you.
Okay, thank you, Francis. We now have two questions from Eloy. Question one is, why should we agree that the conditions responsible for the formation of sedimentary rocks from 3.5 to 3.3 billion years ago are a good indication of what was going on several hundred million years earlier, 4.3 to 4.0 billion years ago?
Well, thank you very much, Eloy, for this fundamental question. Yes, in actual fact, this is very important. The Earth was cooling down. There is some evidence for protocontinents, or that there were protocontinents during the Hadean, I mean, this is this evidence comes from the zircon crystals. And in fact, yes, the early terrestrial crust was quite soft, but even Mike Russell with Nick Arndt published a paper saying that there was a lot of early protocontinental crust, but that was a while ago. So we don't really know, but definitely by 3.5 billion years ago, the crust was slightly more solid because we have the beginnings. Well, first of all, we have preserved crustal rocks. Then we have the beginnings of possibly a very primitive form of plate tectonics. But if there were protocontinents on the early Earth, they would have been pretty similar to the ones that we still see in the early Archean. And I think we have a superb record of preservation. We have a window in time. The reason why the rocks from the Paleoarchean, why they were so well preserved, why the organisms were so well preserved, is specifically because of the high silica content, saturation of the ocean waters. Everything at the water-rock interface was silicified and preserved perfectly. Afterwards, things were not quite so well preserved. So I think this is a reasonable estimation using the rocks from 3.5 billion years ago to suggest that perhaps the situation at 4.2, 4.3 billion years ago was grosso modo quite similar.
Splendid. Thank you, Francis. Part two of this question is, or the second question is, what hard evidence is there to show that these sedimentary basins from the Paleoarchean represent the global climate and are not just local environments? And could we be tricked to make this jump in logic because of the paucity of other well-preserved rocks from this period?
Another excellent question. Yes, we have 2 points in time and space to represent the whole of the Earth's crust. It's not very much. In fact, the fact that there are only 2 tells us that this is quite an unusual situation. But as I said, unusual in the sense that unusual in the sense that they are preserved. But I think previously I answered to the reason why they are preserved. Can we use them as an estimation of the whole of the Earth? I think so. I think based on the modeling of the evolution, geological evolution of the Earth, general evolution of the Earth, I think these shallow water basins, and they seem to be, they're extremely pervasive. Okay, each shallow water basin is different, but in the Barberton Greenstone Belt, in the Pilbara Greenstone Belt, there are numerous horizons, numerous layers, formations, and they all show very similar features. So I think this tells us that we can use these two environments to get a general idea of the early Earth.
Okay, thank you very much, Francis. Our next question comes from Alexandra Zetterlind, and who asks, in the hydrothermal sedimentary scenario, how shallow would the pools be?
Well, from below wave base to the beach, frankly. As I said, we don't have evidence for really deep water sediments. All we can say we have to go on the sedimentary structures. For instance, if we see large wave structures, we know that we're at deeper water depths. All we can say is that there were sediments that were deposited below wave base, and because we're probably looking at protected environments, enclosed environments, we don't really have a lot of evidence for open ocean conditions. So I think, yes, from probably maybe 500 meters to the coastal area, difficult to judge. Depends also on the diameter of the basin, but really not very, very deep.
Okay, and we have one final question from Sigrid Huld, who asks, do you think there are any possible abiotic mechanisms for the formation of microstructures such as the filaments in the biofilms or the pairs of cells in the bead carpets? For example, chemical gardens coated in carbonaceous material with subsequent dissolution of the original abiotic mineral filaments.
This is a good question, and in fact, it's been a question that is always in the back of my mind. But I think it's the fine scale details of and a question that the thing is we know how microbes fossilize. I've been fossilizing them for the last 30 odd years. We understand the mechanism of fossilization, and also exactly what microbes look like. I actually spent 8 years working in a microbiology lab to learn this. So it's the fine scale details that tell us that these are most likely to be microbial in origin. I know in this particular example, and in fact, my PhD student is doing more work on this particular example, the Kitty's Gap chert. We only have morphology. We've got the isotopic evidence. We have evidence from Raman that the organic matter is consistent with the maturity of the rocks, etc., etc. But the fine scale details, for instance, like the meniscus as the cells separate out, and also the fact that the one example where you have a whole cell and then a deflated cell right next to it. Plus, I only showed you a small portion of the images, and there are a lot more. So I think that we can say that there's a strong likelihood that these features are biogenic. We are at the moment intent on obtaining other types of biosignatures, particularly on these features. So it's a very good question. I mentioned in my talk about the paper in 2011 Planetary Space Sciences. I think we discuss it in great depth there. So if you'd like to go read more about the argumentation, please do so. But I think I'm going to have to go back to ExoMars. I'm on the ball now. It's the long-term planners. So thank you very much, Kieran, for fielding the questions and the introduction. Thank you all for listening. And sorry I wasn't with you to give the talk in person, but these things happen.
Okay, thank you very much once again, Francis, and I hope that the simulation is going well so far.